There are three ways by which bacteria perform horizontal gene transfer. These are by conjugation, transformation, and transduction. Today, I will be discussing about bacterial transformation. In the year 1928, a British bacteriologist by the name of Frederick Griffith discovered bacterial transformation in Streptococcus pneumoniae, a gram-positive bacteria. He discovered that a strain S. pneumoniae could become virulent after its exposure to heat-killed virulent strains. In his experiment, he tested the effect of two different strains of S. pneumoniae on mice. The type 3S strain was virulent and caused pneumonia, while type 2R was non-virulent. He injected these strains into mice, and those injected with type 3S strains died while those with type 2R strains lived. He also injected heat-killed virulent 3S bacteria into mice and found that they lived. However, when he injected them with a mixture of heat-killed 3S bacteria and 2R bacteria, they died, although both strains were found to be non-virulent when injected individually. The analysis of the dead mice revealed the presence of live 3S bacteria. He was able to hypothesize that there was a substance present in the heat-killed virulent strain that is responsible for transforming the 2R bacteria into live 3S bacteria. This was what he termed as a transforming principle. Fortunately, in 1944, this transforming principle was identified as the DNA by Oswald Avery, Colin MacLeod, and Maclean McCarthy. By definition, bacterial transformation is the incorporation of a plasmid or DNA fragment from the surroundings into the bacterial cell for subsequent expression upon replication. This ability of cells to take up extracellular genetic material is termed as competence. Thereby, these cells are called competent cells. However, during the uptake, only a 1 to 1,000 chance is present for a successful transformation. Most foreign genetic material that is moved into the cell undergo degradation, failing. Now, transformation may be natural or artificial. Bacteria with natural competence perform natural transformation. In this method, DNA comes from a bacterium that underwent lysis and releases DNA fragments into the surroundings. These fragments come into contact with naturally competent cells for transformation to occur. However, bacteria need to be in a certain stage of growth to be competent. For example, Streptococcus pneumoniae gain competence in their exponential phase where its density ranges from 10 million to 100 billion cells per milliliter. Once competent, they release competence factors to simulate the production of 8 to 10 new proteins needed for transformation. So, transformation begins as a double-stranded DNA fragment from the environment complete to bind with the cell surface receptor of a bacterial cell, thus the uptake is random. Next, the bonded DNA fragments are cleaved by endonucleases into smaller fragments of 5,000 to 15,000 base pairs long. But since the uptake of DNA fragments require energy, only one strand associates with small proteins to move through the plasma membrane and into the cell. The other strand is hydrolyzed by envelope-associated endonucleases for energy. The now single-stranded DNA of fragment in the cell aligns with the homologous region of the genome and incorporates into the original bacterial chromosome through homologous recombination, forming a heteroduplex DNA and completing transformation. On the other hand, transformation in Haemophilus influenzae, a gram-negative bacteria, differs in some ways. Unlike S. pneumoniae, it does not produce competence factors. Also, the DNA complex of proteins are taken in by membrane vesicles. And lastly, they can only take DNA from closely related species. This specificity is caused by the need for the DNA that will be bound and taken up by the cell to have the same 11 base pair sequence that is repeated over 1,400 times as in the H. influenzae genome. Now, the protein complex responsible for taking up the free DNA must be able to move through the cell wall. The complex structure of these protein complexes can be exemplified in the gram-negative bacteria Neisseria gonorrhea. The second type of transformation is artificial transformation. Some bacteria are treated to induce competence because of their lack of natural competence. Two common treatments include electrical shock and exposure to calcium chloride. The application of electric shock to induce competence is called electroporation. This method works because the current allows temporary pores to form that allows the negatively charged DNA into the cell by an electrophoresis-like effect. Chemical transformation through exposure to calcium chloride at 0 degrees Celsius, on the other hand, uses calcium ion to create pores in the membrane. This condition also assists in the binding of DNA to the cell membrane and masks its negative charge. The DNA is then forced into the cell upon heat shock by heating the cells with an additional 42 degrees Celsius thermal current. Both artificial transformation procedures render the cell membrane of E. coli temporarily permeable to DNA. Furthermore, to increase its transformation frequency, strains of E. coli that lack nucleases can be used. It is also easier to transform bacteria with plasmid DNA since they are capable of replicating within the host and they are not easily degraded. To summarize, bacterial transformation is a method of horizontal gene transfer that incorporates genetic material from the surroundings into the bacterial cell. It may be through natural transformation, wherein naturally competent cells randomly bind with extracellular genetic material, or through artificial transformation, wherein cells are artificially induced with competence through electroporation or exposure to calcium chloride to facilitate the uptake of genetic material. Lastly, I'd like to first compare bacterial transformation with bacterial conjugation. Unlike in conjugation, the bacteria do not pick up DNA from direct contact with other bacteria. Instead, they mostly acquire the DNA of dead bacterial cells from their environment. Also, the DNA fragment incorporated into the cell during bacterial transformation is single-stranded.